Hello everyone, in this video I'll be explaining on the topic of gram staining. Gram staining is a staining technique and it is also called as differential staining because this stain differentiate all the bacteria that exist into either gram positive or gram negative bacteria. Coming to the principle here, the principle of this gram staining is mainly depend on the cell wall structure of gram positive and gram negative bacteria. What is the difference in this cell wall of gram positive and gram negative cell wall is the difference in the thickness of this peptidoglycan layer. As you can see here on the right side, which is the gram positive, have a thick peptidoglycan layer, the purple or violet color here, and while on the negative cell wall, the peptidoglycan layer is thin compared to the gram positive. And other than that, the gram negative bacteria has an outer lipoprotein, outer membrane of lipoprotein on the cell wall. And principle of this staining is when the primary stain, which is crystal violet, okay, when the primary stain crystal violet is added, both this cell wall of gram positive and gram negative bacteria get stained and appear violet in color. After that, when grams iodine is added, which is act as a mordant or act as a binding agent of the stain to the cell wall. Okay, so when the grams iodine is added, it binds the helps or enhances the primary stain to bind better to the cell wall or the cell wall of the bacteria. And in case of gram positive, since they have a thick layer of peptidoglycan with numerous teichoic acid cross-linking, which results in a better binding of this crystal violet, and grams iodine act as a mordant and trap the agent, act as a trapping agent and increases the affinity of the cell wall for a stain by binding to the primary stain. And then when the decolorizer is added, on the third step, when the decolorizer is added, then since the decolorizer is an alcohol base, in case of gram negative, the decolorizer when added, it will dissolve the outer lipoprotein membrane. Okay, the decolorizer when it comes in contact with the gram negative cell wall, they will dissolve the outer lipoprotein membrane and washed off, okay, thereby exposing the thin peptidoglycan layer. This is in case of gram negative bacteria. So when the decolorizer is added, since the decolorizer is alcohol based, it will go and dissolve the outer lipoprotein membrane and then exposing the thin peptidoglycan layer. And since the peptidoglycan layer here in case of gram negative is a thin, so the crystal violet stain which has entered inside will leak out. They will leak out because of the thin peptidoglycan layer. So they will get leak out and washed off. They will get washed off and get decolorized. All the color gets removed. This is in case of gram negative bacteria. While coming back to the gram positive bacteria, here when the decolorizer is added, which is an alcohol base, then it will dehydrate this peptidoglycan layer. It will dehydrate this peptidoglycan layer and thereby all the pores of this peptidoglycan layer gets tightened and then not allowing the stain, the crystal violet stain which has entered inside to come out. So the primary stain crystal violet get trapped inside and then they don't get decolorized. The stain does not come out even after the addition of the decolorizer because the decolorizer will dehydrate the peptidoglycan layer, thereby decreasing the pores and trapping the primary stain inside. So the gram positive will remain as violet in color and remain undecolorized. Next, in the final step, when the counter-stained safranin is added, here in case of gram-negative, since now they are colorless, when safranin is added, they will take up this safranin stain and appear pink in color because the safranin stain is pink or red in color. While in case of gram-positive, since they are already colored with crystal violet stain and appear violet in color, they will not take up any more stain and then they remain as it is, violet in color, because they have take up the primary stain. So this is the reason behind why gram-negative bacteria appear red or pink in color and gram-positive bacteria appear violet or in color. Okay, so this is the principle of gram staining. Coming to the procedure, the requirements of this test are: first is take a clean glass slide, then take a spirit lamp in order to sterilize the inoculating loop while making the smear, and then staining rag is required. Pasteur pipette is required for transferring the volume of staining reagent on the glass slide smear. And then finally microscope is required to observe for the observation of the smear. Reagents that are required for gram staining are: in case of primary stain, it is crystal violet. Mordant is the gram iodine. Dechlorizer acetone alcohol, and secondary stain or counter stain is safranin. So coming to the procedure includes 4 basic steps of the gram staining. Here first, after you have prepared the smear, place the smear on a glass slide, contain the smear onto the staining rag. After that, with the help of Pasteur pipette, transfer a few volume of the crystal violet stain and pour it over the smear and cover the entire smear. Keep it for 1 minute. After 1 minute, take the glass slide and gently wash under running tap water. After that, with the help of Pasteur pipette, transfer a few volume of grams iodine and cover the smear and keep it for 1 minute. After 1 minute, discard off the grams iodine stain and then dechlorize for about 10 to 15 seconds, drop by drop, and then wash it gently under running tap water. And then place the slide back to the staining rag and with the help of Pasteur pipette, take a few volume of the safranin and pour over the smear, cover the whole smear and keep it for 1 minute. After 1 minute, wash it under running tap water and then allow the smear to dry at room temperature properly. After the smear get dried properly, then you can proceed for observation under the microscope by placing 1 drop of oil immersion and then observing under 100x oil immersion objective lens. When observing under the microscope, what is seen? Here 2 types of bacteria will be seen here: gram-positive bacteria and gram-negative bacteria. In case of gram-positive bacteria, they will appear violet in color, while in case of gram-negative bacteria, they will appear red or pink in color. And then not only the differentiation of gram-positive and gram-negative bacteria, but based on their shape, they can also be described into either cocci or bacilli. So here on the left side, the bacteria shape is round shape with violet in color. So they are gram-positive cocci in clusters. Why mentioning clusters? Because they are arranged in grape-like cluster in group. While on the right side, you can see the bacteria are pink in color with rod shape. So they will be called as gram-negative bacilli. And then what errors can occur during the procedure is mainly on the decolorization step, which is very crucial. If you have over decolorization, it would in this case gram-positive bacteria may appear pink in color, and in case of if you have not well decolorized your smear, then gram-negative bacteria may appear violet in color.